Trevor.
So it's an honor to be introducing Trevor Price today. When the grad students were asked to select a speaker, we were told to pick somebody who is pushing the field of evolutionary biology forward in innovative and challenging ways. And for once, it was easy for us to agree that Trevor's work certainly does just that. I think the broad nature of Trevor's work, which focuses on speciation and biogeography, but also on things like sensory ecology and animal behavior, made him very appealing, not just to MVZ students, but also clearly to students throughout the campus. He's also a particularly fitting candidate because his academic parent, Peter Grant, along with Rosemary Grant, visited the MVZ as last year's student-invited speaker. So, as a mammologist, I'd like to pretend to be disappointed that 4 of our 5 speakers have now been ornithologists, but with people like Trevor coming to visit, it's hard to be too disappointed. So Trevor did his PhD with Peter Grant, then at the University of Michigan. He then moved on to a position at UCSD before moving to the University of Chicago, where he is now. I'm not going to embarrass Trevor too much by mentioning the number of papers he has with over 100 citations, and the number of times he's published in journals like Science and Nature, but I will say he's famous for designing some truly elegant studies that use very thorough and integrative fieldwork to address major questions in evolutionary ecology. Trevor is a true field biologist. He's worked all over the world, and I think that probably gives him an advantage for our MVZ custom of mentioning a fun fact about each of our invited speakers. It was hard to pick just one for Trevor. He provided a few, but I think it's fitting to mention that he has one of the few tenable excuses for not finishing a field season. He and his colleagues were captured and taken hostage by Kashmiri militants in the early 1990s. Reasonable excuse, I think, before they could study the last of the 8 species they intended to work on. And I actually went online and found a few interviews you gave about this story. It is a truly terrifying story, complete with AK-47s and hand grenades and a miraculous escape in the middle of the night. I highly recommend listening to those interviews. So luckily, that experience did not deter Trevor from continuing his work. So we're very fortunate to be able to welcome him here today to talk with us about sensory drive, color, and color vision in birds. So please welcome Trevor.
Thanks very much, Tali, and thanks for inviting me. I think the very first seminar I gave after I graduated was here. That was a long time ago. And I remember very well being extremely nervous, and 10 minutes into my talk, my mouth went completely dry. My tongue stuck to the top of my roof. And, I think everyone in the audience noticed. And I feel pretty nervous again today, actually. For some reason, or maybe for a good reason, for the last few years I've been sending out quiet vibes saying, I really want to come to UC Berkeley because I think it's doing the most relevant work to what I'm interested in. So it's a tremendous honor to be here. And if you prefer, I'll be happy to talk about the hostage story now instead of this. There's a good saying though, because Peter and Rosemary Grant, I guess, were here last year talking about the Galapagos. One of my first pictures of the Galapagos. This is Natasha Bloch, who was a student of mine, sitting on the Galapagos. And I bring her up to begin with because a lot of the ideas and work that I'm going to be talking about were done in collaboration with her. And carrying on the Galapagos theme, here's a couple of Darwin's finches, which are not colourful, dull, boring birds, but I use them to illustrate 2 points. One is that most of the work that I've done uses the comparative method, which seeks to explain adaptations in terms of correlations of traits with environments. And so we know we've got a big finch and a small ground finch here, and we know why they're big and small, because the big one eats big seeds and the small one eats small seeds. So this comparative approach to interpret traits is a very powerful method. The second thing that I want to point out about these is that they're very different in size. This is a young adaptive radiation where you see diversification in traits directly connected to ecological niche. So when a lineage gets to a place where there's very few species at present, you see diversification to fill these niches. So with those 2 points, now let's move to continents. which are much older environments. And you can see the 2 points I just made sort of don't apply. This is the 4 species of bellbird, with the females are green and the males differ in these secondary sexual traits. And they're ecologically very similar to each other. It looks like they're occupying essentially the same niche in different places. And so the argument that I would make is that all the niches have got filled up. We're at the last stage of an adaptive radiation where you have ecological replacements that are diversified in sexually selected traits. The second point, coming back to my previous slide, is it's extremely difficult to think about correlates of the environment that have created this diversity. It's very hard to use a comparative method here because we can't think of environmental differences. And in fact, this idea that you get diversification in the absence of environmental differences goes way back to the Reverend Gulick, who studied snails in Hawaii. And he wrote this paper in Nature, which said, I've accumulated a large body of facts indicating that separate fragments of a species, though exposed to the same environment, will in time become divergent. And Darwin made a couple of points. He sort of disagreed with this. He said, first of all, we don't know that the environments themselves are not different. There'll always be small differences. And secondly, you haven't got a mechanism. So Gulick got a second paper in Nature, back in 1890, where he said, yeah, we do have a mechanism, it's yours. The preferences of the sexes for certain shades of arrangement then become slightly different. So if we look at the bellbirds again, the idea would be that in different places, different mutations would have cropped up that females liked. And with no gene flow across the system, you don't have homogenization until you've got enough differences cropping up in different places that they just don't recognize each other as suitable mates. So if this is what's going on, it's extremely difficult to explain diversification in color or any other secondary sexual trait. It just depends on mutations. But there's a growing body of evidence, right, that ecology does affect evolution of color. And I'm going to be talking about this today, even if most of the diversification in color that we see is because of these arbitrary divergences. If we can explain some of it in terms of ecology, then we'll be making an advance in understanding this puzzle of why species differ in color. And it might turn out, when we get far enough along, that we can explain a lot in terms of ecology, but we're far from that. So I've given this introduction several times. You know, for example, why have you got 3 wattles here and say a blue face here? Well, some bright student in the audience suggested, well, maybe there are just worms in this place. Females like worms, they get tuned to worms, and then these guys make worm-looking things. So there's an example where ecology might actually be driving this diversification. We're far from understanding that yet. But what I want to talk to you today about is specifically about color, about how color may have diversified in the face of environmental differences, and how I feel we can make advances in this area by not necessarily just looking at color, but looking at the perception of color, getting a mechanistic understanding of how perception of color might change, and how the environment might influence that. So that's the focus of the talk. So just going back to this idea of arbitrary divergence, which we think might account for a lot of this. When sexual selection was resurrected from a dormant period in the early part of the 20th century, we had a bunch of models turn up that could explain why you would get arbitrary diversification. And I just want to introduce, I think probably the most useful models are these neural net models, which I won't talk about, but I'll talk about the more famous one, which is the runaway sexual selection model that Landy developed, because I'm going to talk about developments of this model later. But under this model, as I'm sure you're all familiar with, the idea is that in the absence of any environmental differences, you could have one population or species where females were having a low strength of preference for a male, and another population or species where they had a long strength preference, and you would, and these positions are arbitrary along this so-called line of equilibrium. I just want to make it clear what I mean here by an intensity of preference. A simple way to think of this is it's the number of males a female will search through before making a decision that she's going to choose the one with the longest tail. So if she only searches with the first one, takes the very first male she got, then there's no strength of preference, and the tail evolves to some natural selection optimum. But if she'll search through 20 males before choosing the one with the longest tail, there'd be a strong preference. Males with long tails would have lots of offspring. You'd end up with a balance here. So this was a mechanism for arbitrary diversification, but I think you could immediately see that if she has to search through 20 males here or one here, there's plenty of opportunities for ecology to influence the costs and benefits of search will affect where you evolve to. So we can already see how in these sexual selection models, ecology will affect, how much you'll prefer different males, and we're going to talk about how it will affect the kinds of males that you might see. So I think for evolutionary biologists, the ideas about how ecology might affect the evolution of colour stem only back to Endler in 1992, where he introduced this term sensory drive, and he really emphasized how perception of colour will change against different backgrounds, making it easier for females to distinguish males in certain colours depending on the background. And so that you should therefore get environments driving colours in different directions. And the most recent summary of this work is Ryan and Cummings. And subsequent to that work, there's been quite a large number of studies that have actually used the comparative method successfully to show correlations of colour with environment. And what I want to do first in the talk basically is take you through a comparative study that we did that invokes changes in social selection pressures with environmental differences. Then I'm going to talk about how natural selection pressures might create perceptual biases that drive colour. And then I'm going to talk about colour perception more mechanistically and how we can look for comparative associations of colour perception across environments. So, make it sort of historical. There are only 6 species here because 2 of them could not be studied before the infamous event. But there are 8 species that live along, of old world warblers, that live along this altitudinal gradient in Kashmir. This is the place that we were forcibly removed from. And we started to study these 8 species quite a few years ago now. And our main motivation for studying them was their colour patterns. They're pretty much the same in colour, but they have a diversity of colour patterns, so that this species basically has an eye stripe and an underpart, and so does this one. But this one, you notice, has an extra wing bar, and this one also has an extra wing bar. Well, this one has 2 wing bars and eye stripe. Now we're getting really psychedelic. This one has 2 wing bars, eye stripe. That's a crown stripe. And this is really getting crazy here. Underpart, 2 wing bars, and a rump patch. And this one, 2 wing bars, crown stripe, rump patch, and you can't see it very well, but white in the tail. So we had this nested pattern, which meant that we could sort of unequivocally say that this was the brightest bird in the system, and this was the dullest bird in the system. So each of these patterns is formed, just saw this on my thing, that each of these patterns is formed by an absence of melanin in the tip of the feather. And every pattern is basically the same. And further, so here we've got a crown stripe on this species and a crown stripe on this species. When you make the phylogeny, you find that aspects of the pattern have evolved multiply. In particular, the crown stripe has evolved here as well as here. So that if you're worried about comparative methods in phylogeny, which I'm not, but if you are, we've got at least a couple of times when the things evolved twice, so we can use some of these comparative methods. So Karen Marchetti, my partner in Escapeology, set out to do her PhD thesis investigating why it is that different species have different colour patches. It's actually kind of an interesting story because I was a disciple of Landy. I worked in India, so I was a disciple of Gandhi, and then I worked here and a disciple of Landy. And Landy, I was so influenced by Landy that I thought these colour patches were a classic example of movement along a line of equilibrium. So that we basically had more patches, more search, less patches, and it was just drift along a line of equilibrium that was driving it. Fortunately, as we all know, graduate students don't listen to professors, and so Karen came out and basically started from scratch. The first thing she did was take this one species here, which has 2 wing bars and nothing else, and this species sets up its territory in the birch trees before breeding starts. When it snows, it just drops down the mountain and waits for the snow to go away before it comes back up again. But it's a very easy species to study because you can catch it really easily on the snow, and then you can walk around and map its territories. So this is just a map of territories in the days before GPS. Eventually the snow melts and the females come along and build their nest on the ground, and so this would be the territory of one of these males. So the first thing she did was to paint these males, and she could either make the wing bar bigger or she could paint it out completely, and it worked painting it out completely because when you paint it out completely it looks like the bird without any wing bar, and fooled us all the time. It didn't fool the other birds because of presumably vocalizations and other things, but it fooled us. And when she did that, she found this striking result, which is the birds with bigger wing bars got bigger territories. The birds with reduced wing bars, some of them lost and they got smaller territories. And this sort of thing has actually been, although I thought it was pretty remarkable, it's been repeated in other species like red-winged blackbirds as well. So we're arguing that these colour patches are a target of sexual selection through male competition. She further showed that these colour patches are used in male-male display by doing playbacks. So this guy with a crown patch comes in and shines its crown patch. This one, I'm not going to do it, turns around and sticks its bum out. And clearly these patches are associated with male competition. But the main result for today's talk is there's a strong correlation with habitat brightness. So the species that live at the bottom of the mountain in dark habitats tend to have crown stripes and rump patches. The species that live out in the open with no patch at all tends to have no patches except the eye stripe. This species is pretty interesting. It's the one with no extra patches. It lives on the edge of the forest though. So it actually is sort of an example that proves the rule. And although I was still skeptical, I finally got convinced because the one species that's missing from here is the one that breeds in rhododendron, in the really dark rhododendron. And when you see it, all you sometimes see is the white tail feathers flying around in the rhododendron. So I got pretty convinced about this. So the results of all this was that we concluded that there's a balance between conspicuous and cryptic as set by the light environment. And in fact, these patches have got more and more complex as it got darker or lighter. So that result of Karen's led me and Dolph Schluter to build some theoretical models of what might be going on. And we just took Landy's model and we modified it to, first of all, assume that females were getting benefits from choosing different kinds of males within a species. So basically the sort of handicap model, a male in high condition is providing some benefit that's being indicated by its trait. So, we built the full-fledged model, but the bottom line is that the female intensity of the female preference should evolve in response to the environment. The average female preference is proportional to something we call the detectability, how easily the trait is seen against background, times the honesty, the benefit the trait is giving to the female by her mating with a male with an exaggerated version. So, for example, if there was a trait that indicated parasite load in one environment and not another, then you might expect this to become more exaggerated in that environment. But detectability, Endler's big point, has a bigger effect than honesty. In fact, when you look across environments, this is just thinking again, going from a darker to a lighter environment. Imagine tails are more detectable, a difference between males is more detectable out in the open than it is in dark when you're talking about tails. Then what you see is this sort of exponential increase in, or a large difference between populations and the exaggeration of the male tail as you move between environments. The reason for this is that females that can easily distinguish males for the same amount of search are getting more benefits when they can easily distinguish males than when they can't. So there's direct selection on the female preference to increase. But the males as well, by being more detectable, are getting more often chosen when they've got a long tail. So you have a double whammy where the female is selected to search, for the same amount of search, females get a bigger benefit, so there's evolution of the preference to search more. And the males get a bigger benefit when they're out in the open, so there's more selection on the male trait. So what this says is that you really should see big changes in socially selected pressures and socially selected traits when you move from one environment to another. And although this Karen study applied this to a light-darkness gradient, it should also apply to color background gradient. So that's, you know, an argument that as the environment changes, you should see evolution of female perception and especially male traits. And it comes from the evolutionary literature, it comes from Landy's original models. By contrast with that, and with a much longer history than Endler's, sensory biologists have argued that natural selection pressures in the environment, predators and prey, cause evolution of the sensory system, and then male traits evolve to take advantage of that, or to exploit differences in female perception that have evolved because females are now under selection to see different things in the environment. Although sensory biologists don't trace their history back to Darwin, Darwin, of course, as always, said it first. It was in a pretty obscure place, but I finally found something. Darwin said it may be suspected that even the habit of viewing different coloured surrounding objects would influence their taste, being female. So this is the idea, is that something about prey or predation drives you to change your perception so you can see these things more easily, and that then influences the kinds of males you like to see. So this idea has been applied fairly extensively now, particularly in the aquatic environment, where you have a very steep gradient, not only in light intensity, but in light quality. As we know, we filter out the reds and the UVs, and you end up with just blue light once you go down a few meters in the ocean. So these are two examples, because the first one I put up here because this was by Helen Rodd and Co in Toronto. The popular accounts of this said they call it guppy love. And she noticed that if you drop an orange peel, which she did, into the river, the guppies come crazily towards it. If you drop an apple peel into the river, they don't. So she realized that the prey, which I guess is sort of tanniny and orange, was really attractive to guppies, and argued that male guppies have become yellow to exploit this preference that evolved for prey capture. The second example from Terai and Seehausen concerns cichlid fish, where it's not prey, it's light environment. And in this muddy cichlid Lake Victoria waters, there's preponderance of red light lower down. And these two cichlid fish, one is blue and one is red, and what they did was they looked at the opsin sequences, and then they measured opsin tuning and showed that the particular opsin they studied was more tuned towards red lower down and towards blue higher up. And they argued that this tuning had occurred to enable females to see prey in the prevailing light environment. So it was the light environment, not the colour of the prey. and that tuning had then been taken advantage of by the males to make themselves more conspicuous to females given their tuning. So these are two examples of natural selection, one for prey, one driven by the light environment. And as I said, it'd be mostly applied in the aquatic environment. And I introduced the sea housing example because what we set out to do was use these similar methods across birds in the terrestrial environment. Now, terrestrial environment obviously differs in colours of prey, red berries or yellow bananas. They don't vary as much in the light environment as they do in the water, of course, but they do vary in their light environments. So the question is, does this variation in light environment influence female preference? That's what we're going to be talking about. So what we've got here is a wavelength of light going from UV up to red. And then this is the number of photons, if you like, coming from a small point. And so if you look at the sun, it's pretty well white, equal across the whole regime. If you look at blue sky, you've got a lot low wavelength light. And if you look at leaves, you've got a lot of green light. So, one of the questions will be, could these light environments affect tuning, and could that affect colour? I won't be able to answer it. It's a question for future generations probably, but anyway. So what I want to do before I get to that then is introduce you to some of the basics of colour vision, which I found to be extremely difficult to understand. And the first point I want to make is that colour vision depends, colour perception depends not only on the let me make this slightly bigger. It doesn't depend only on the wavelength of light hitting your eye. It depends on the background against which you're viewing the colour. So there's considerable scope for phenotypic plasticity, if you like. If you're a bird and you move into a different background, you can actually change the way the colour is being perceived. And to prove this to you, I have here a bowl of fruit. And I have here the same bowl of fruit with a blue filter put over these three pieces of fruit. And I have here the very same bowl of fruit with a blue filter put over the whole lot. And if you focus on the pepper, if you will, it's red, blue, and what is it there?
Red.
Well, no, it's not. Oh, yes, it is. No, it's not. Watch this space. So that's the red bit. And I will just move it up, or I won't move it up. It's turned blue. So it's exactly the same wavelength of light hitting your eye there as it was there, but the complete blue surround here has made you perceive that as red. It turns out, people who work in human colour vision know this, but it's always pretty interesting to show it to, people like us. It blew me away when I saw this. But I think a lot of avian colour vision has evolved to make this as red as possible when viewed against a background that changes. What's actually happening is there's so much blue light hitting our eye that our blue cones, which I'll talk about in a minute, are getting desensitized. And the way we perceive red is not by just the amount of light that the red cones capture, it's the ratio of the red to blue cone capture that matters. And I'm going to go through that now anyway, if I can not screw this up too much. So, don't look at this. Don't look at that. Crap. Hang on a sec. Sorry. Although I've just shown you the environment can have a huge impact on the way we perceive colour, we also know that genetics can have a huge impact too. And if you were just viewing a scene that was just one colour, there was no background around it, that would be entirely determined by the the physics of the light and the mechanism of color perception in the retina. So we are trichromats, and in our retina we have 3 different cone types concerned with detecting color. These cones are tuned to catch different wavelengths of light. So we've got a short wavelength cone, a medium wavelength cone, and a long wavelength cone. And yeah, so the short wavelength's capturing, the blue light. The green one is capturing green light, the red one's capturing red light. There's considerable overlap here. What actually happens in the retina is, as I've already said, is we take ratios in the retina, and the brain receives a ratio of this to the sum of these two. And when the ratio is very high, we view it as blue, and when the ratio is very low, we view it as yellow. And it also gets a ratio of this to this. So if there was no blue in the system, and you had a ratio of M to L that was high, you'd see it as green. If you had a ratio of M to L that was low, you'd see it as red. Those ratios are taken in the retina and sent up to the brain. They're not taken in, they're not processed in the brain. You send up these 2 ratios. We're trichromats. Very few, no mammals except a bunch of primates are trichromats. Nearly every mammal is a dichromat with just an S and an L. Mouse actually is UV shifted in the S and it has an L. There's 1 or 2 mammals that are monochromats, but like I say, a few primates like this mandrill, and some, this is my joke, and some even more distantly related primates are trichromats, but beyond that, every other mammal is a dichromat. So the cool thing about this is that the tuning is affected by this visual pigment termed an opsin. And for example, in humans, this 30 wavelength shift between the peak of the green and the red is determined by 3 amino acid substitutions. And what you can do nowadays, of course, is express this in vitro and measure the green, measure the wavelength of absorption. You can express this one in vitro and measure its wavelength of absorption. And you can do the genetic manipulations to ask how each of these amino acid substitutions are affecting the tuning. So it's a beautiful example where you can translate genotype directly into phenotype by using this in vitro model. Now in primates, I want to now just talk about the positions of these 3 peaks within primates, because what we're going to do now is say these positions are adjusted to features of the environment, and then we're going to switch to birds and ask, when we look across birds, are features of these adjusted to the environment? So primates have been the best studied, so why is it that we've got the blue here, we've got the green here, we've got the red here, and why indeed is it that we've got 3, whereas other mammals have got 2? So the green, a really good explanation for why we've got a lot of M and L in this region is because of us growing up in a strongly green environment. I told you already that there are 2 channels that measure color that send up to the brain. There's a 3rd channel that just measures light intensity and movement, and that's the sum of these 2. So for example, if you're looking at a green leaf and a bird flies across, you don't compute the bird's color. You want to observe its movement. And the best way to do that is to have maximum input to the cones of the color in the background, so that as the bird moves across, it contrasts most with the background. Okay? Because if a blackbird comes across, you're just cutting out all the signal to the cones of that, and that'll give you the biggest contrast with the cones that are still receiving the background. So it's reasonable to think that these are tuned here to just measure movement, because we know it's the sum of these that is used to measure movement in general. I haven't told you this, but I'll tell you now. 90% of the cones in the eye are the green and red ones, and that's because they're involved in movement and detecting blacks from whites. So that's the L plus M. It might be reasonable to think that the blue is about the right place to detect movement against a blue sky. Pretty speculative, but reasonable. And that's this ratio of the S to the L plus M. Okay, so if you had a red bird flying across, well, let's not do that. Let's not go there. And then finally, and more interestingly, is why are the L and the M this ratio, and why are the L and the M so close to each other? And the argument here is that with this spacing, you're best able to detect orange fruits against a green background. And you can show, if you think what we're looking for is a ratio here, if you have a background that gives you a ratio here of about 1 to 1 in the catch, okay? And then you move to here, you have a ratio of cone catch that's almost 10 to 1. So over this region, you have very fine discrimination by having these two with narrow overlap. And that's why, that's thought to be why these two are so close together, because this region here is when you just move slightly, you move a big change in the ratio, and it's the ratio that's being sent up to from different parts of the retina that the brain is comparing. So we've got the evolution of colour vision that we suspect is related to this ability to see orange fruit against a green background. And, of course, primates are the most colourful mammalian group. We would argue that the evolution of colour vision in primates has subsequently led to exploitation by males, if you like, of the sensory system that's evolved in response to natural selection pressures. So that's the basic sensory drive argument. But this is very general. It's basically said primates have taken advantage of this, and of course they could have done this if every primate had exactly the same colour perception system. You could get this arbitrary diversification in colour, just like we talked about at the beginning with the what were they? Bellbirds. So what we want to do is ask if perception varies across species in a way that can be related to colour and environment. And to do that, we turn to the New World warblers, which, like lots of things in the New World, and unlike lots of things in the Old World, I'm English, in case you hadn't noticed, I'm bright and interesting, not dull and boring. And so, for interesting reasons, I'm sure I won't have time to get into, but I'll be happy to discuss. The New World warblers are all diversified in colour, and they're dimorphic, so females are often dull and males are bright. And we've got blues, we've got reds, we've got yellows, we've got carotenoids, structural colours, and melanins creating in colour. So we set out to ask if diversification in the visual system could be somehow related to this colour and environment. So what about the visual system of birds? Well, unlike mammals, unlike primates, birds are tetrachromats, and they can see in the UV as well. So there are 4 different cones associated with colour in the retina. Here, for example, is a green, this is a UV, this is the blue, and this is the red. And you may have noticed that they're actually colourful, the cones, unlike in humans. And the reason for that is there's a filtering pigment there that takes out low wavelength light, sharpening the sensitivity of the cones. So we've filtered out, you can see the blue and the UV are wider than the green and the red because these pigments are filtering out the lower wavelengths of the green and the red. I think that's got something to do with this business of colour contrast and the fact that you see a blue the same against different backgrounds, but other people think it's for other reasons. So this compares a typical bird with a human. You can see there are narrower ones, there are 4 of them, and we can see into the UV. So I'll just get that one. So So we set out to look at these. We measured sexual selection by simply using a sexual dichromatism score that someone else had done. So some species are monomorphic, would have a dichromatism score of 0, whereas the black-throated blue warbler is highly dimorphic, and this yellow throat in the middle is moderately dimorphic. So we have a measure, if you like, of intensity of sexual selection. We don't have a measure of colour that we've been able to successfully use yet, but we do have a measure of intensity of sexual selection, which I'll be talking about. The next thing to do is to look at the opsins. And to do that, you walk around Chicago in the fall or the spring, you stand under a building, bird drops out, thank you very much, I'll have his eye. It's not really that funny. 600 million birds a year die hitting buildings in the US. 2.4 million birds a year die by being eaten by cats. I used to make a joke about that, but then a few people in the audience would always walk out because I was being so bad about cats. Cats are the big killer, but buildings are number 2. And so actually to illustrate just how big the bird death is, we wanted to study 14 species of New World warbler, 2 males and 2 females from each. We had no trouble getting the sample. There was only one species where we got 1 female from. So that, I think, illustrates just, the mortality that's going on. Oh, it's 16, sorry, not 14. So for the 16 species, we we got the transcript, we took the RNA out of the eye, made cDNA. Then we went to Belinda Chang's lab in Toronto, it's a bit of a royal we here, and measured you can just basically, grow the thing up in a bacteria, put it into a human cell, do a whole bunch of stuff and put it into a spec and measure the opsin absorption spectrum. So here's Natasha doing that. You have to do it in the dark. So there's a little bit of red light. She's here. This is not a colour vision test. I won't cut it out and she'll suddenly appear. So anyway, she looked at all 4 of these opsins, and I'm not going to be able to discuss all of them today. I'm just going to focus on the blue, SWS2, for the rest of the talk. The bottom line is that when you sequence these opsins, they don't differ very much among species. The amino acids differ, but when you express them in vitro, you find the wavelength of peak absorption does not vary very much. So what I've done here is I've plotted wavelength of peak absorption for all the 16 warbler species against their habitat as a measure of light environment. So I think, the ovenbird, this dimorphic undergrowth species, actually has the most red-shifted opsin, and it's living in the undergrowth where the blue and the UV have been filtered out. So when the blue and the UV are shifted out, its opsin seems to have been shifted. But the shift is only a couple of nanometers. And we've got polymorphisms in humans of 6 nanometers. So there's almost no correlate. There's not much opsin evolution happening. But there have been a bunch of other studies of opsin absorption spectra, not based like we did using in vitro expression, but using microspectrophotometry of the retina. And this is not as an accurate method, but we added all those species to the system. And you can see now that understory species do tend to be very red shifted, whereas open species tend to be blue shifted. And this would be in accordance with the prevailing background in these 2 environments. So we've got something like the red-billed Leothrix. This is a Himalayan bird. It's damn weird that anyone looked at its retina, but it turns out it was a captive bird in Germany. And this is one of the Astrodid finches, and you can see that this has got a very blue wavelength, is very short wavelength shifted opsin, and this has got a long wavelength shifted opsin. So this is the first evidence from a terrestrial environment that the light environment can affect opsin tuning. We've got plenty from the aquatic environment, but it's been hard to get it from the light environment. And the reason seems to be, well, first of all, from the terrestrial environment, the reason seems to be, first of all, the light environments don't vary as much as they do in the aquatic environment, but also because opsin evolution is incredibly sluggish. It just doesn't evolve very fast. We've actually got old world warbler sequences and new world warbler sequences that are completely identical despite 30 million years separation. So there are other aspects of the opsin system that might evolve more quickly, and of course the classic one would be expression levels. Are some opsins expressed more in one species than another? So we set out to measure opsin expression and found out that indeed you have much more evolutionary lability in expression levels than you do in tuning. It can vary up to 15 times between species, but what was rather remarkable was that it was sexually dimorphic in expression. So what we found actually is that there's a correlation in females between the amount of opsin expression and how dimorphic the species is. So males that are highly dimorphic, species are highly dimorphic, females have lots of opsin expression. I put this slide in first because it doesn't look, as far as we can put foraging height in, which I showed you before was affecting tuning when you looked across a bunch of species, and it doesn't seem to be mattering. So it looks like, if this is some measure of intensity of sexual selection on this axis, and this is trait on this axis, it almost looks like Landy world. In fact, male opsin expression doesn't correlate with dichromatism at all. So when you put these two together, you can make a landscape model. This is opsin expression dimorphism. And this is plumage expression dimorphism. So the ovenbird would be somewhere about here. It's got this, males and females are identical, and they have very similar opsin expression. Whereas the black-throated blue is the most dimorphic in color, and it has the most dimorphic opsin expression. So, does this indicate arbitrary divergence? I'm very reluctant to say that. I believe that there will be natural selection pressures that we're missing that are somehow driving us along this line, but we've got absolutely no idea what they are now. That's future work. But at the moment, we can't reject the good old Landy model, which is what I had in my mind 30 years ago when we started working on the Philosopher's. It would be extremely interesting to know, of course, or we'd like to know, what the hell it means to be expressing more or less opsin in the eye. We haven't really got much of a clue. But what an increased density of opsin does is it increases the width of this capture. You can think that if the opsin was so dense in the cell, it would capture every photon, and the width of this would be flattened. So the more and more opsin you add to a cone cell, the wider this gets. And as this gets wider, of course, it's creating different overlaps between other cones, which I showed you when you looked at the red and the green in primates, is very important because that gives you the place where you can most easily distinguish slightly different colors. So one model is that increased opsin expression will affect color perception by changing overlap between cones. So that's about it. I've told you that we've got this theory now about how color perception and color should diverge across environments. I just want to leave you with this lasting thought that the more and more we understand about how color vision works, and then we can apply that in a comparative approach, I think the closer and closer we're going to get to understanding just how much the environment contributes to color divergence. I don't know if we're ever going to understand this without including models of mutation and development. And maybe mutation and development is explaining 90% of this, but it might be that 10% of this is indeed explained because there are only worms here. And with that, I'll thank you very much. The next slide shows. I believe it is. I'll just show you this one slide. So, the New World warblers are part of a 900 species radiation that originated about 20 million years ago as a result of a flock of birds flying across the Bering Strait. 900 species from this flock. When that flock flew across the Bering Strait, it was a finch. And finches live out in the open, where they tend and communicate using colour. So I believe it's just a historical legacy of passing through a finch-like form on the way to becoming a New World warbler. John Endler disagrees with me. He thinks that this is a younger group, and so they've evolved into an environment that had less selection, less parasites and predators, and less costs to become colourful. But I think it's just historical contingency.
Do you find any variation on the filter based in broadening or thickening those absorption patterns for the options? The filters. So you talked about.
Oh, you mean the oil droplets?
Yeah.
Yeah. Everyone talks about oil droplets. Oil droplets are extremely difficult to measure, but Matt Toomey has recently done some really interesting work. He's at Washington U, where he's starting to do this now, and he showed that when opsin tuning moves, and we show it can move quite a bit when you look at extreme species, the filtering moves with it. But actually showing that the filtering, which would seem a logical way to change the width, varies across these species, no one's attentive because it's so difficult to measure accurately oil droplet concentrations at least. But what Toomey showed was that the actual filtering molecule had changed, allowing them to track the movement of the opsin tuning. That's all I can really say at the moment.
Yeah, so I guess if you have increased opsin expression in a particular color channel, then you could either increase the opsin packing, as you talked about, or it could be increasing the relative number of cones in that channel. Can you distinguish that in that system? And have you guys looked at that?
Well, we did. We saw the, I showed you that. You probably know the answer to this, right? You're seeding me here.
No, I don't know.
I showed you that picture of the rattle thing. So we looked at 3 or 4 species and did cone counts, and they crudely correlate. So the red opsin, across opsin classes they correlate. So the red opsin is the most highly expressed, it has the most cones. But we could find no correlation across species. So we believe that it's change in opsin density. That, of course, is assuming that RNA translates to protein. But at the moment, we can't. We don't think it's cone density.
So these breed seasonally, presumably, and so does the female opsin expression vary seasonally?
So we can collect them in the spring and the fall, and we found no significant effect of the season. They probably vary diurnally as well. I mean, we collect all these at 6 or 7 in the morning, but I think a large number of transcripts vary diurnally. But we could find no effect of time of day or season.
Similar question, but were these all sexually mature birds? And for juvenile ones, did you treat those separately, and did the effects, the dimorphism and option expression still?
Good question. So most of these were, I just don't remember. Obviously in the spring, they're all adults. In the fall, they'd be a mix, probably quite a bit of juveniles. And I honestly don't know. I don't think we ever looked at the difference. All I would say is that we didn't have a difference between spring and fall.
With the looking at the degree of sexual dichromatism and then opsin expression, so it looked like you just said opsin expression, or were there any differences in the different types
Oh, the
opsins
cones?
being expressed? Yeah.
Yeah, so this is all, sorry, everything I talked about was with the blue, SWS2.
Well, that was all SWS2. Okay.
So we've got it for all the others, but it just gets too complicated to talk. So for example, this is the UV, and you still have a dimorphism, and you can see for UV there seems to be a correlate with ecology. And we found the same also for the green, some correlates with ecology. But, and it's always a dimorphism in direction of females a little more. The red, LWS is hard because the long wavelength is also expressed in these things called double cones, which I didn't talk about today. These are used for motion detection, completely different system, so it's hard to separate them. But the others, dimorphism is present everywhere, and there may be some ecological correlates we're picking up. We need to do more work on this.
Other questions? Well, if not, I'll remind everyone there that Trevor's giving another talk tomorrow at 9, and let's thank him again for a wonderful talk.